So you all probably know Dr. McLaughlin is out at a conference this week. So my name is Elliot. I'm going to be leading the lectures for today and then also on Thursday, picking up where she left off with y'all, looking at the neural basis for cognition. So I have the attendance sheet being passed around, so that's all going to work the same. Make sure you grab that before you leave. And I guess also be forewarned, I'm getting over a cold. I went and rode my bike in the cold rain this weekend, so my voice might go, and I have a feeling we may not get to everything that we need to today, so we'll just pick it up again on Thursday when I rest a little bit more.
Are you following her slides or these your own slides?
Mostly hers. I've added a few things, and so those aren't available online yet, but I'll make sure that by the time I'm done, she'll have those for you all, but they're pretty much the same. So just briefly, I'm going to talk about my background so you guys understand why I would be here talking about neurocognition, or the neural basis and cognition. We'll briefly review what Dr. McLaughlin has already gone over with you so that we're kind of primed to build off of that, and then also to make sure that I'm on the same page as you all. Then I'm going to briefly go over kind of a strategy for navigating the brain. You know, we're going to be looking at a lot of different sections of the brain, and they all have these really weird, convoluted names, and really if you break them down, it's much easier to understand what those mean, and that'll help you a lot as you start to learn about that more. Then we'll talk about specific areas of the cortex, one of the kind of, think of it as kind of like the home of what we think of as cognition overall. And then we'll talk about brain cells, how those go into all of that, and then finally we'll start to get into the visual system, which is one specific way that we can look at how the brain relates to function. So really briefly, I said my name is Elliot. I got my degree in psychology at UNCA on the other side of the state. Minored in neuroscience and did some research and courses in computer science. So I especially focused on cognitive psychology, so all of that really ties in well with what we're talking about today. So I've always been interested in how the brain kind of basically works as an information processing unit. So it takes all this data information in the world outside and it converts it somehow, and then our thoughts and our behaviors come out of that. And a lot of that relates to ideas from computer science too, which I think we'll talk about later in the course too. But currently I'm a PhD student here in the Human Factors of Applied Cognition, which is the same area that Dr. McLaughlin is in. I teach introductory psychology, but it's an online course, so it's been a while since I've had the opportunity to have a real lecture with this many students. And then what I'm studying right now is decision-making, expert decision-making in wildland firefighting. And y'all will learn more about decision-making later on in the course too. But I really like this. One reason is because I get to do, part of the year, my homework takes place out in the woods, and then part of the year I'm stuck inside the lab, but I like that I get to mix it up like that. So, starting to review about what you already went over with Dr. McLaughlin, you started talking about Capgras syndrome, right? And that was something that we used as an example of how people have used all the different methods we've talked about to determine that Capgras syndrome, which is, you can recognize faces of people that you know, but you don't really believe that they're the people that you know. You kind of think that they're imposters or something. And so after people have studied it in a lot of different ways, we found out that this ability to recognize faces as people that you know is actually all these different sub-cognitive abilities, like it has to do with factual memory and visual matching and emotional connection, and that all of those sub-components of cognition are actually handled by different areas of the brain. So it's just combining the fact that we know that all these different abilities take place in different parts of the brain and how we kind of study that, all the different methods that we use. And then another example of that was the classic example of Phineas Gage, who, as you all know, he lost part of his frontal lobe in this railroad accident, and then after that he lost specific cognitive abilities that relate to like inhibition and emotional regulation and things like that. And we'll talk about the frontal lobe and its functions a little bit more later. So, because he lost that very specific part of the brain, but not all the other parts of the brain, he only lost very specific functions. And then you went over one of the kind of broadest ways that we break down structure and function of the brain, which is looking at it as the hindbrain, the midbrain, and the forebrain. And so the hindbrain is kind of the basic survival instincts, like regulating heart rhythm and blood pressure. And that's something that we share with a lot of other organisms too, so it's like a very basic evolutionary structure. And then above that is the midbrain that has some more complicated functions like coordinating movement and relaying audio information to different parts of the brain. And then finally the forebrain, which is really what we usually talk about when we talk about cognition. This is where the cortex is, like I was talking about, it's very important in cognition. And then some subcortical structures like the hippocampus and the amygdala. Hippocampus is related to forming new memories, and the amygdala is related to emotions and emotion regulation. And then another way that we can organize all the structures and functions was looking at the cortex specifically. We can organize it into these 4 different lobes. The frontal lobe, and then that's separated from the parietal lobe by this fissure, the central fissure, and then the temporal lobe down here, and occipital lobe back there. And we'll go over some more about their specific functions, but I think you all touched on them some, and you probably just kind of know them as common knowledge to some extent. And then this idea that rather than just being organized by lobes in each hemisphere, it's also that each hemisphere actually handles different functions. So in general, a lot of the perceptual processing that goes on, like sense of touch and sense of vision, those are handled by the other side of the brain. So if you feel something on your left hand, that sensation is actually being processed in the right side of the brain. So in general, we have this lateralization, and when you talk about the information being processed on the other side of the brain, that's contralateralization. And then you talked about all these different methods that you can use to image the brain. You know, if we want to relate how the brain relates to function, we use all these different imaging techniques to see what's actually going on inside the brain. And you talk about all the different pros and cons of it. A lot of it comes down to the fact that certain techniques can be used to look at structure, certain techniques can be used to look at neural activity, and then all of those have different resolutions in time and space. So if you want to look at a very specific area, you would use something like fMRI rather than EEG. But if you want to look at a very specific point in time, you would use something like EEG. And so I think this is where you all left off, the idea that because of all these different pros and cons of all these different imaging techniques, one of the things that we do is just use a bunch of them and look at the same problem. Is this the last slide that you all saw? Yeah. Okay. So yeah, this specific example, we're talking about using, like I was just alluding to, EEG is really good for looking at what happens in the brain in very short spaces of time. But fMRI is really good at looking at what happens in the brain in very specific spatial locations. So if we can use both of those, then we can start to get a really clear picture of what the brain is doing when you perform certain cognitive functions. And so one of the examples that they use in the book is this study by Tong and colleagues. And they were looking at 2 very specialized regions in the brain. One is called the fusiform face area, or the FFA, and that's really active when people look at faces, especially when they're trying to recognize a specific face. And then they compared this with another very specialized area called the parahippocampal place area, the PPA. And this one they saw is really active when people look at places, especially like in the study it was pictures of houses. So when people look at pictures of houses, the FFA does not get excited. And when people look at pictures of faces, the PPA does not get excited. So they have very different roles and they react very strongly to these very specific stimuli. So they first identified these areas by using fMRI. So they showed people a picture of a face, and this FFA region lit up really strongly when they did that. And then they showed them pictures of a house, say, and these PPA regions lit up very strongly. So now we were able to pinpoint very specific locations of the brain where these functions take place, but they wanted to know how that happens over a very short span of time, because these images are derived over something like 6 to 10 seconds or something of taking pictures of the brain. But we know that when you look at a picture, or look at someone's face, it doesn't take you 6 to 10 seconds to recognize that face. It happens much faster. That visual processing happens much, much faster. So EEG is a good way to look at something that happens that quickly. So now, since we knew exactly where we wanted to look, the big, coarse spatial resolution of EEG wasn't detrimental. We could place the electrodes right where we wanted to look and then start looking at very short spans of time. So in this study, they use what's called, what is it, binocular rivalry? Yeah. So they show a picture of a face to one eye, and they show a picture of a house to the other eye. And they tell people to, so they're able to see the visual information of both these different objects at the same time. And they told people to press a button when their attention turned from either the house to the face or back the other way. And they were able to measure the electrical activity that was happening in these areas that they wanted to look at when people chose to focus on the house or focus on the face. So they started by focusing on the house, and then they started paying attention to the face more. This blue line is the activity in the FFA, that very specialized facial recognition area. And the red line is the activity in the PPA, the place-specific area. So you can see that after zeros where they push the button, they said their attention changed. You see this huge spike at the FFA because it's starting to process a house, or a face, sorry. And then you see a huge decrease in the PPA because it's not paying attention to a house anymore. It's not having to do that processing. And then you get the exact opposite effect when they were looking at a face or attending to the face and then decided to attend to the house instead. You see the place area gets more active and the face area gets less active. So one thing that I thought was interesting here, and I don't know how much you all tend to participate, so if there's silence, that's okay, but there's a huge gap right here where they start attending to a face, and this one goes way down. And here, they're attending to the house, but it's a much smaller gap between them. Does anyone have any ideas of why there's that difference in the activity levels? Yeah?
Recognizing a face might be more important to survival.
Yeah, definitely. That's the general idea. So face recognition is something that we know is critical for survival in our evolution because we're very social creatures. So you want to be able to quickly recognize who someone is and maybe what their emotions are, reading their face. And while recognizing a place is important, it maybe doesn't have to happen as frequently or as quickly as you do for faces. And the FFA actually is apparently a really good specialization area. So there are a lot of experts that are really good at bird watching and identifying different birds just automatically, super quickly. And it turns out that they're actually doing that processing in the FFA region too. So it looks like that this particular area is set up to be able to specialize and be really good at something really quickly. And so that speaks to how important the recognition of faces is for us. Oh, and so it's important to note that, they were, like I said, they were presenting these two different images to each eye. So they were receiving visual information of a house and a face at the same time. And when that was happening, there was no difference in these two regions. So the critical difference is that when you're attending to one or the other, even though that same information is hitting your eyeball, it's the processing that happens after that that's important in these two areas. And so the book also talks about that just because we see activity in these regions when we perform certain cognitive functions, we can't necessarily say that those regions are what are responsible for those cognitive abilities. We can't, it's correlation, but we can't say that it's causation. So another way that we have been able to determine when certain areas are causally related to certain cognitive abilities is by looking at patients with lesions to those areas. And so we've seen that in the FFA specifically, that when people have lesions in the FFA, they lose the ability to recognize faces. And that's called face blindness or prosopagnosia. So we have a short clip about showing someone that has face blindness, and also Groupon. And so this person doesn't have damage to the FFA, but it lets you know what prosopagnosia is.
What they have trouble doing is putting the features together into a gestalt, in a kind of a whole. A person with prosopagnosia will be able to see this as a face, be able to recognize that it's made up of different objects, but he won't be able to identify this face as unique.
I probably always had an awareness that I didn't recognize faces very well. I would say that I see it holistically. I'm told that subconsciously I break it down into geometric shapes. I basically have categorized people. There's certain numbers of faces in the world, and I automatically put somebody in a slot. There would be middle-aged, balding man, slim, attractive, dark-haired women. I group them together and automatically put them in one area.
No one really knew about developmental prosopagnosia until about 20 years ago. These individuals have difficulty recognizing faces from the time they're born.
Well, there is my cousin's wife, who I've known for 25 years, and at a family function, I had no idea who she was.
Can you recognize yourself in a mirror?
I scare myself sometimes, but I do recognize myself.
As far as we know, there's nothing obviously wrong with their brains. There may be some subtle problems that they have, either in connecting the regions that are necessary for face recognition with each other, or in having those regions smaller.
And growing up, I was best friends with two little twins that no one in the neighborhood could tell apart, except me. And I think it was because I knew them so well that every little nuance of their face was something I had memorized.
Not only is it inherited, but it's a dominant trait. It's carried on a dominant gene.
My brother suffers from the same thing, and he's very friendly. We tend to say hello to everybody.
Do you think that your sense of a good-looking woman is the same as your male?
I suspect, yes.
You're a photographer.
I'm
Wife's face cut out of a magazine. You would know it was her. If I was to leave and to come back with another woman, do you think you could distinguish my face from hers?
absolutely.
Let's test it out.
Bring it on. It's tougher than I thought. And I'm going to say Hillary is the one on my left. I would say that that is Hillary. Mostly I think it's Hillary because of the smile. And I think I remember the smile.
You're wrong, Jeff.
I can't believe it.
So boring.
I can't believe it.
Oh.
Something about the music and the like camera movements in that, like bring me right back to 2007. It's like a real world or something. So, like I said, this particular individual didn't have damage to the FFA that we know of, but if someone did, this is what would manifest behaviorally. So when we have cases like that, where we have someone who has damage to a very specific area that we know has a very specific function, or at least that we know has activity that relates to a certain function, if that area is damaged and so is that cognitive function, then we can start to say that they're causally connected. Does that make sense? Or ramble for a second? Cool. So we don't always have that benefit, though, of having patients that have very specific lesion damage like that. So we have this other technique called transcranial magnetic stimulation. Have you all heard of this or seen this before? So this is really one of my favorite methods, I guess, if you can have that. Watch another couple short clips here. Basically, the idea is that it's this device that creates a magnetic field, and as we know, magnetic fields are related to electrical fields somehow. I don't know exactly the physics. But what ends up happening is that that magnetic field disrupts the electrical activity of the neurons under the wand that they use. And so that causes disruptions in function there.
If we start at the back of the brain, we've switched off my visual cortex, so made me unable to see things. If we move a little bit forward in the brain, we've interfered with people's ability and my ability to see faces. A little bit further forward, we've interfered with the ability to perform actions. Is that moving your finger? Quite odd. It's not your finger. It's not my finger anymore. We've interfered with the ability to compute with numbers.
6, 7, 8. Oh, yeah.
We've interfered with the ability to speak.
London Bridge, London Bridge, London Bridge is falling down.
you think of something you can do, and we've basically interfered with it with this machine.
2, 3, 4, 5, 6, 7, 8, 9.
So what's the problem?
I don't know.
So I really like that clip and TMS in general because, one, the things that they were asking him to do, move his hand and sing London Bridge, which should come pretty naturally to a lot of us, those abilities were completely disrupted. It's not like he wasn't paying attention closely enough or something, but yeah.
Does that hurt? You mean I can hurt?
From what I hear, it doesn't hurt because it's not actually electricity, like like they were shocking you with something. It's just a magnetic stimulation. So, as far as I know, there's no pain that's associated with it. Also, as far as I know, there's no lasting effects. That's obviously something that they're still exploring because it's pretty new. But TMS is actually being used to treat depression. I'm not sure exactly how they use it, but apparently it's becoming pretty clear that it's kind of revolutionary in treating certain kinds of depression. Yeah?
I had a question about face blindness.
About what? Sorry.
Face blindness.
Uh-huh.
So what he did, he would like memorize the geometric shapes on people's face.
Mm.
So if somebody's like just not good at overall recognizing faces, right? Like if you know that you have met this person in your class, whether you don't remember their name or whatever, but does that mean that the same type of area is affected? Would you say that person has a certain degree of face blindness? But like, if you're better at recognizing people and places more than another person, does that mean that they have less face blindness than another person?
Yeah, so her question was kind of looking at face blindness as we saw it there and how it's related to the FFA, but there are varying degrees of face blindness, like maybe some of us just individually are not as good at recognizing or remembering faces or recalling what their name is or something. And so wondering if those are really the same thing. And there's definitely a possibility. I think that you would have to look at each case individually and start to break it down kind of in the same way that we've done here. Maybe you could compare the activation and the FFA of someone who just in general is not as good at someone else at recognizing faces and compare that with someone who's really good at it or someone who has FFA damage and see how those compare to each other. But there are a lot of different cognitive processes that would go into that, and it might be working memory, or it might be how you define features of a face. And I think that the feature definition seems to be what's related to the FFA here, because like you were saying, he takes these very coarse, like wide-angle features, like brown hair, bald head, maybe glasses sometimes, whereas most of us might be able to, not explicitly, but maybe we're using cues like length of eyelashes and stuff that he hasn't built into a feature like that. So it would definitely, I think it would depend case to case, but I think I wouldn't be surprised if in general our overall ability and the individual differences in that ability are related to maybe the organization of the FFA or something like that. So another thing I wanted to look at in this video is coming back to the idea of contralateralization. So remember I was talking about that sensory processing happens on the other side of the brain from the body where it's happening. We see the same thing with motor control too. So going back to this video where he's moving his left hand, right? Or left hand, correct? So where are they placing the wand? Is it on the left or right side of his brain? Yeah, so it's on the other side of his brain, and that's affecting the movement in his left hand.
Quite odd.
It's not your finger
It's
anymore.
not my finger anymore.
We've interfered with the ability to...
And then this is actually the same person, but from a different video. This time, he's doing stuff with his right hand.
Okay, so writing.
So he's using his right hand, but look at where they place the TMS wand. You see, he's still, with his left hand, he's still able to hold the clipboard, but the TMS wand is on the left side of his head this time. So that's just an example of contralateralization in motor control. Yeah?
I Does it affect everyone generally instantaneously, or is there people who have some kind of resistance to it? Sometimes it can delay, like this guy's like happens now, but something will maybe go to last a second or 2 seconds or anything like that with this method?
Sorry, can you say it again?
So when he gets the magnetization right to the spring, it instantly changes his actions.
Uh-huh.
So I'm asking is there people, I know it's new, but that have like a delayed reaction to it yet, or is there people who this isn't really affecting?
So the question is, one, how instantaneous is that effect of the TMS, and does everyone experience it instantaneously? And then two, sorry, what was the second part?
Basically just that.
Just that,
people
okay.
have resistance to it.
So, as far as I know, it's always instantaneous like that. And so the idea is that they're putting the wand over the part of the brain that is basically, and we'll review this, it's called the primary motor cortex. And it's basically where the signals to move your hand first leave the cortex and start going out somewhere else. So if the neurons in that specific area lose the ability to send that signal, which happens immediately because it's basically happening at the speed of electricity, then it will instantaneously affect you.
So it's kind of intercepting that signal.
It's creating interference on the process or the signals being moved by the neurons. So then overall, what we're getting to here is the importance of the idea of localization of function. Certain specific areas of the brain tend to have certain specific abilities associated with them. They handle very certain cognitive skills. So in the book, they have these pictures. Yeah. They have these pictures where they're looking at the difference in brain activity as people look at images of objects or scenes and when people imagine those same objects and scenes. So on the surface, we would think that those are two pretty different cognitive abilities, but when you have people do this in the fMRI machine, you see that there's actually a lot of overlap in the areas where these, where you see heightened activity while they perform these cognitive skills. And so they're obviously not exactly the same, but there's apparently enough like statistically significant overlap to say that they're kind of the same process at some level. So even if we don't see them that way, if behaviorally, they at least start kind of using the same processing and same brain areas. And so another example of this, we'll go back to the TMS video. So right here, obviously they want to disrupt his motor control, so they have placed the wand over the motor cortex. And one of the important parts about that is that as he's struggling to move his hand, he's still able to speak.
We've interfered with the ability to
Did y'all see that?
numbers.
He didn't struggle to speak while he did that. But now they move it, it's still on the same side of his head, but they've moved it maybe a few centimeters, and he's no longer over the motor cortex, they're over an area that's affecting language specifically. And so we can't see his hand there, but I don't imagine his hand is jerking like that still. And so it's just another good example of how very specific areas have very specific functions. So that kind of catches us up and wraps up what Dr. McLaughlin was going over with y'all. Do you have any other questions before we start to move on into looking at more specific areas of the cortex? Cool. So yeah, the big takeaway there is localization of function. Yes?
So, here's everything, the TMS. If someone had like brain damage when they were younger and like this plasticity of the brain, and like localization in different areas, would TMS be functional in different areas then? Like for like the lower cortex, that like that's actually really that's actually about it.
Yeah. So she was asking about the idea where, especially with kids, because they have greater plasticity, if they suffer damage in a certain area, other parts of the brain will take over processing that the specialized area would have done before. And so if that's the case, then would TMS affect them differently? follow-up question is, could TMS then be used as a diagnostic tool? So like if you expect that using TMS in this certain area would affect their ability to move their hand or to speak, but you don't see that, then obviously it's being handled somewhere else. So maybe you could say, oh, they must have had damage there at some point when they were younger and it got co-opted by another part of the brain. So while that's fresh on my mind, yes, I do think that theoretically that's a really interesting idea. I've never heard of that before. I don't think that they would do it because they still don't really know what happens if you shut off the wrong part of the brain. So unless they're really sure that this is where they should be disrupting function, they probably won't do it. So then related to that, and the first part of your question, where do different people, is it handled by different areas? Yeah, and so one of these videos is when he first comes into that lab, they actually do some sort of other imaging technique, I think fMRI, or maybe MRI, just to make sure that the structures that they're looking for are really where they expect them to be. And also everyone's brains will have slight variations in the way that they're laid out. So you want to identify that specifically for each person before you start using TMS on them. Any other questions? All right, so localization of function and all those different imaging techniques and the pros and cons of them, and we can use them to relate brain structures and behaviors. So now I wanted to, this is something that I've added in, so I don't think it would be on a test or anything at this point, but it was really useful to me as I started to learn about neuroscience and cognitive psychology. And that's what I alluded to is a lot of these brain regions have these really convoluted, kind of intimidating names, but they all kind of share the same vocabulary. So if you understand that vocabulary, it becomes much easier to quickly recognize where they're actually talking about in the brain. So they've used some of these in the book before. I think there was a little table at one point that broke down some of them, but when if you're looking at this side view here, and you're looking at the front of the brain and the back of the brain, they often refer to those as the anterior for the front or the posterior for the back. And so those are pretty easy to remember because posterior is like your butt and anterior is opposite of that. So if you just remember posterior, that's the back of the brain. They're also broken down into the top or the bottom of the brain, which is either referred to as superior or dorsal for the top and inferior or ventral for the bottom. And so the way that I remember those is superior, higher, so that's the top, or dorsal, like dorsal fin on a dolphin or something. And then if you look at it from this perspective, you still have the anterior and posterior for front and back, but you also have lateral and medial. So lateral, like the side, you see that in a lot of different contexts, like football and the lateral fissure over here. And then medial is pretty easy to associate with being the middle. So when different brain regions that we look at have these words, then you can start to figure out where they actually are spatially. And then especially when we pair it with what we've already talked about, the names of the different lobes here, and use other features like the fissures. We have the central fissure here, the lateral fissure. You'll start to see all of these tied in together and like grouped into one weird, confusing name. But if you break down into all these different features, you can actually make sense of where it is. Another useful thing is that the cortex is all wrinkly and pruny, and all the bumps there are called gyri, or gyrus singular, and then the valleys are sulci, or sulcus singular. So you'll see those pop up, and if you can start to look at all those together, then you'll start to understand where things are. So as an example, we have this really convoluted term called the ventromedial prefrontal cortex. This is really important. They found in things like decision making, which you'll talk about later, executive control, which you'll talk about later, and different parts of attention, like that comes up in mindfulness research a lot. And so if I just looked at that right off the bat, I don't know where that is, but I'll start with usually looking at the lobe that they're talking about. So in this case, frontal. So I know where the frontal lobe is. I can stop looking at all the other parts of the brain here. So we know it's somewhere in here or in here. And then prefrontal means, the pre means in front of, basically. So we're going to look at the front part of the frontal lobe. No, first we're going to look at medial. My bad. So medial means middle, so you're looking at the middle part of the frontal lobe. And then we're going to look at pre, which is the front half before the rest of the front. And then finally, ventral means bottom, so we'll just look at the bottom part. And that's where the ventromedial prefrontal cortex is. So the name can tell you exactly where things are, and once you start to understand where these different structures are, and people are telling you what cognitive abilities are related to those areas, then you can start to see how they're related to other ones that you've heard about and other abilities that you've heard about. And so this is where they first start to break down the cerebral cortex into some of the structural organizations that we'll get into now. So this is out of your book. First, we're going to look at the sensory and motor projection areas. So I talked, when we were looking at TMS, I talked about the primary motor cortex, and sometimes that's called the primary projection area or motor area. But basically what it comes down to is these are the areas in the cortex where the information or sensory information first comes into the cortex from other parts of the brain, and where signals related to motor behaviors first leave the cortex to other parts of the brain. And then after those primary projection areas, we'll talk about the rest of the cortex, which we call association areas. So starting with these primary motor projection areas, like we saw were being manipulated in the TMS video, what you see, if this is a cross-section of the primary motor projection area. And what they've done basically, really simplified, is they, sometimes they did it exactly this way, but if you can stimulate really specific sections, like basically poking it with a little electrode or maybe even just a little needle, and then see movement in different body parts, then you know how much of that cortex is responsible for moving a certain body part. And what you see is that areas that need really fine motor control, like the fingers and hands or the lips and mouth, those have greater representation on the motor cortex. So there's basically more neurons devoted to handling those motor signals because you need more control there, so you need more processing power there. And then for areas like the toes, where we don't need much fine control, there's very little area on the motor cortex that's responsible for handling that. And so the primary motor projection area is the posterior frontal lobes. And so using what I was just talking about, again, we can look at the frontal lobes specifically, so we know it's up here somewhere, and then looking at posterior, meaning the back of it, so it's the back of the frontal lobes. So even if you just had the name, you could imagine where that is. So it's this thin sliver here, that's the primary motor cortex. I won't go into the TMS video again, but you can see that where he was placing it specifically was pretty much right over these areas that we are seeing in the diagram. So that primary motor projection area, like I said, is where the output for motor behaviors first leave the cortex. Primary somatosensory projection areas are where the sensory signals from somatosensory specifically talks about touch, where those first come into the cortex. So these are the anterior parietal lobes. So again, parietal lobes right here. Anterior is the opposite of posterior, so it's the front of the parietal lobes. So it's this blue sliver here, right next to the motor cortex. So that's the sensation of touch, mainly somatosensory. There are other projection areas for auditory information. That's mostly handled somewhere in general in the temporal lobes. It says superior, so it's like the top of the temporal lobes here. And then primary visual projection area is all in the occipital lobe. That's something that we're probably familiar with. Occipital almost always means visual processing. So that's where those visual signals first enter the cortex, is back here. And then similar to how we saw greater representation in the motor cortex for body parts that need greater motor control, we see a similar proportional representation for body parts that need greater sensation, specifically sensation of touch. So the primary somatosensory projection areas are on both sides of the brain here. And again, areas that require very precise sensation. Oh, I'm sorry, this is actually motor versus somatosensory. Anyway, so things like the lips and face and tongue, where you get a lot of sensation, those have a larger representation, and so more neurons basically handling that incoming sensory information. And here's the auditory cortex, so that's the primary auditory projection area, and again, primary visual projection area. So again, this is one of those contralateral things too. So there is a somatosensory area or projection area on either side of the brain, and the information that gets processed on either side comes from the other side of the body. So this is actually looking at auditory information. So if you hear something from your left ear, then it will be processed on the right side of the brain. If you hear something from your right ear, it will be processed on the left side of the brain. And the same thing for touch. If you feel something with your hand on the left side, or with your left hand, it will be processed in the right side of the brain. And if you feel it on the right side of your body, it will be processed in the left side. Get it? Vision is a little bit different. So by the same logic, you would kind of expect that anything you see with your left eyeball would get processed on the right side of the brain. Anything you see with your right eyeball would get processed on the left side of the brain. But it doesn't actually work like that. Instead, it's anything that you see on the left side of your body. So this is called the visual field, everything that you can see in front of you. And if you draw a line straight down the middle of that, anything that appears on the left side of your visual field is processed on the right side, so on the other side of the brain. Anything that you see on the right half of the visual field is processed on the other side of the brain. So even though both your eyeballs are seeing the same image, they're processing it in the same area. This has to do with this diagram, which I believe is in your textbook. And it's kind of weird to break down, so I'll just go over it quickly, but you understand the general concept. Things on the left side of you get processed over here. This kind of explains why that happens. So basically, in each eye here, on the retina, which is where all the cells are that actually receive light and begin that sensory signal, the ones on half of it, from left to right, on half of it, they'll come back here, and they'll go straight back here to this side of the brain. The ones on the other half of the retina will actually cross over at this place they call the optic chiasm, and those end up going over to the other side of the brain. And then vice versa on this side. Some of them go straight back, the ones on the other half of the retina go over to the other side. Then they hit this LGN, and then obviously primary visual cortex, that's our primary visual projection area. So, summary of the cerebral cortex. Again, there's the localization of function. The contralateral aspect is really important, both for sensory information and motor information. And depending on how precise you need that motor or sensory information, you'll have greater or lesser representation on those projection areas in the cortex. So any questions about any of that?
If you're blind in one eye, would that affect the visual cortex that's attached to that eye?
If you're blind in one eye, would that affect the visual cortex attached to that eye? Yes. Let me try to think of how I would expect it to affect it. So basically, that one eye would be sending information to both halves of the brain, right? To both halves of the visual cortex, because that one eye can see stuff over here or see over there. So if it's, I guess it would depend on what the source of the blindness is, but I'm going to assume it's some physical dysfunction in like the eye itself. That information just won't move at all from here to the brain. So what the brain might do is use that unused space in the visual cortex for some other function, like we were talking about with plasticity. But yeah, in general, for that form of blindness, it's just the signal would never even get back to the brain. There are some forms of blindness where one of those connections, either the one that goes to the same side of the brain or the other side, will get disconnected. So you still get some visual information from an eye, but you only get it from half of what you actually see. So yeah, all different forms of blindness, but those are some ways it might manifest. Anything else? Okay, so those were those primary projection areas. And those make up about 25% of the brain. So it's these, this is the motor projection area, here's somatosensory in red, here's the visual, and then they don't label it, but auditory is in here too. So it's a very small part of the brain. The rest of it is what we call these association areas. And this is more what I tend to think of as cognition usually. It's, one, our memories tend to be stored in these association areas. So, you know, with the projection areas, it's like taking information in and changing it into something else. With the association areas, with memory, it's just storing that information. And then with association, it's being able to combine different pieces of information into different concepts. And this is most of what the cortex does. So when you have damage to areas of the cortex, you get all these different behavioral disorders. We'll look at some clips of these. What time is this class now? So a lot of these will look pretty similar, but we can talk about the ways that they're actually different. But this first one is called apraxia. And so in general, it's loss of the ability to initiate voluntary action. And that usually for us means initiating a voluntary movement. So it's like you want to make a movement, but you're not able to. So this particular example has to do with the movements
Larissa
involved
and Scott
in
Bilker of Barnegat, New
speech.
Jersey, have good reason to worry about their son, Brandon. At 8 years old, Brandon is unable to speak intelligible, complete sentences. He suffers with a mysterious neurological disorder called verbal apraxia.
Early on, we didn't know if Brandon was a late talker. You know, everyone told us, oh, you know, Einstein talked when he was 5, and he'll probably speak just fine.
I pledge allegiance to the flag.
As we watch Brandon and his 6-year-old younger brother, Grant, recite the Pledge of Allegiance, it is apparent how much effort and focus it takes for each word Brandon repeats to come out properly.
One nation, one heaven, under God, indivisible, indivisible.
Children who are verbally apraxic have been misdiagnosed as mentally retarded or autistic.
Lori Roth is a speech therapist with a specialty in oral motor skills.
The pathway from the motor center of the brain does not have a clear path to the muscles of the mouth.
So she just highlighted there what distinguishes this, this particular form of apraxia, which is that the person understands words and they know what words they want to use, but the motor signals leaving the motor cortex don't quite make it to the mouth or the vocal cords. So you can see the child was able to say stuff, but it was taking a lot of effort, it was very forced. But so this apraxia is related to just being able to enact, like basically put your thought into action. You still have the thought, you just can't make it happen. So this is agnosia. And this is, agnosia in general is loss of the ability to identify or recognize objects. Sometimes it's face agnosia or name agnosia. In this case, this guy has object agnosia.
I'm going to give you a choice, and you tell me what this is.
Okay, I can tell you
Okay. Is this a clock, a lock, or a telephone?
A lock.
Is it a lock?
A clock, a lock, or a telephone? Okay, let's rule out. Let's rule out a telephone. What? A clock or a lock? Clock. That'll be a lock. Must be a lock there.
Are you sure?
No, I'm not. But I don't recognize it as a telephone.
Mm-hmm. Do you recognize it as a lock?
No, honestly, I don't. But it's just, it really looks very indescript to me.
Uh-huh.
So in that case, somewhere in his brain, he knows what a clock and a lock and a telephone are. And he's probably able to perceive all the visual information on whatever picture he's being shown really well. But there's damage somewhere in the cortex where those ideas are supposed to be combined and then also matched to a name. So it's like a conceptual understanding of that object, the perceptual information that you have in the object you're trying to identify, and the name of the object. Somewhere those aren't able to be combined. So that's one of the things that the associative cortex does. And looking at agnosia again, I said it's in general this inability to recognize or name, mainly recognize objects. And so that's where prosopagnosia, which we talked about, face blindness, this inability to recognize faces, at the end of that word is agnosia. So it's, I assume prosopo has something to do with facial recognition. So two more aphasias, so aphasia, these are two different forms of aphasia because they affect very specific regions of the cortex. One is responsible for generating language, and one is responsible for understanding language. So these are the Wernicke's and Broca's areas, and so even though they both relate to language, you'll see the way that they manifest behaviorally is very different. This is the Wernicke's aphasia, which is just an inability or a decrement in the ability to understand, basically, language.
Hi Byron, how are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people over them over there. They're diving for them at the moment. They'll save in the moment. He'll have water very soon for him. With luck for him.
So we're on a cruise and we're about to
We
get to
will store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment they don't show a darn thing.
With the iPad that we were doing. Like here?
I'd like my change for me and change hands for me. It would happen. I would talk with Donna sometimes. We're out with them. Other people are working with them.
So this Wernicke's aphasia is really interesting to me. One, it seems different from the others because everyone else obviously was experiencing some sort of frustration and awareness of their behavioral deficit. And to him, he doesn't seem to notice or care. And even to us, if we don't pay attention to the words he's actually saying, it sounds like real speech. And that will differ greatly from when you have damage to the Broca's area, which has Broca's aphasia.
How about your writing? No. What about your writing? No. You haven't been affected? No. It's affected. It's affected? Yeah. Okay. What's wrong with your writing? Right.
So this one, I think the biggest difference that we can look at is from him and the kid in the first video, where the kid in the first video obviously had the words, but he just struggled to make them happen. This individual seems to be struggling to get the words into his consciousness, basically. It seems like sometimes they are almost there, then they kind of slip away. And so these are all, at some point, I think we'll see pictures on the cortex of where the Wernicke's area is and where the Broca's area is. And you'll understand the differences between damage in those areas and how you get these behavioral deficits. Yeah?
With the Wernicke's aphasia, are the words that they're saying similar to the ones that they want to say? That's what I've heard about that, or is that?
That's interesting. And again, I assume it differs from patient to patient. And so I don't have a clear answer, but I'm going to ramble for a second to see if something coherent comes out. But one of the reasons why we kind of recognize it as almost real speech is the flow and the tone and maybe the sentence structure too, to some degree. Like, he uses nouns and verbs in a lot of the right places sometimes. Now, whether those nouns and verbs, how similar those are to what he wanted to say, I'm not sure. Yeah, I'm not sure how that works. I'll try to read up on that for next time, and maybe we can look at it in depth a little bit more. So, again, what we're looking at are these association areas. So you can tell that different pieces of information in different forms are there for these people, but they're not able to combine them in the right way. And depending on which association area is damaged, their ability to combine those things is different too. So just a couple more. Here's neglect syndrome. So this is where you get information about the whole world, but you don't notice it for half of the world. So a lot of times this is visual, and so a person can technically, they have all of the structures there to see, to receive visual information from the whole scene, just like you and I would, but on one half of the visual field, it just doesn't register for them. And it's not like, and they don't notice it either. So what ends up happening is they behave as if that half of the world, it doesn't exist. So here, both of these examples are dogs. So this dog had a stroke, and obviously is completely ignoring half of the food bowl. And I wondered about this because, like I said, it's usually visual neglect, but obviously dogs rely on their sense of smell, and he doesn't even seem to notice that they're over there. So I'm not sure if it's all of the sensory information that is kind of ignored on one side, or maybe it's, since visual information is usually such an important cue, if we don't see that over there, then we'll even ignore all the other sensory information, because it's like, if we don't see it, it doesn't exist. Even if I think I smell it, it doesn't exist, I don't see it. So another dog, although now realizing that's kind of sad, but I think this one is apparently one of Dr. McLaughlin's friend's dogs or something. But yeah, just perfect division right down the middle of your whole world is just not there. I'm sure the dogs are still happy. Yeah. Yeah, and so for someone